Hello, welcome to the virtual tour of the Cima lab. My name is Sydney, an HST student in the lab, and I'll be your tour guide. The Cima lab is an interdisciplinary group that develops diagnostics and treatments aimed at improving human health. Our group is led by Professor Michael Cima and consists of grad students and postdocs with diverse scientific interests. Our research projects are divided into groups based on clinical area or technology. Each group meets weekly with Professor Cima. Day-to-day work in the lab is project-dependent, but is always exciting. I'll let some of my lab mates share more about the projects they are working on.
Hi everyone, I'm Erin and welcome to the Cima lab. Today I'll be talking about the Injector Subgroup, where we work on building tools to sample proteins from the interstitial space, which we then use to study neurologic disorder. So what does that mean? Here are paraneurons. We often think of neuronal communications as a one-to-one electrochemical message, wherein information is directly passed from one cell to another. In actuality, this is much more complex. Messages are dumped into the space surrounding the cells, which can signal many other cells. In disease states, these messages build up over time. So you're left with this diverse signaling landscape filled with protein and genetic material and few tools equipped to study them. In the Cima lab, we're currently working on methods to sample from the space to track how proteins in the brain change over the course of disease. We're doing this by building tools such as neural implants that can interface with house-made pumps. We then perform analysis using mass spec to understand the peptide landscape. In addition to the brain, ISF exists throughout the body, so we've begun sampling from 3D tissue cultures in order to see how proteins change during organoid development. Thank you all for listening. Feel free to reach out with any questions.
Hi, I'm Max, a fifth-year MEMP in the Cima lab. I work on two main projects. The first is on the Injector project, which you'll hear about from other students, and the second is the in situ drug sensitivity screening project. In that project, we use a micro-device implant, which is only 7 millimeters long, and is filled with 18 different chemotherapies, microdoses of each. This device can then be implanted into tissue, for example into a tumor, for 24 hours, and over that time the drug diffuses out of the device into the surrounding tissue. We can then take the tumor out and look at how the tumor responded to each drug individually using immunofluorescence staining. This allows us to see in a short time frame what drugs may be effective in treating a certain tumor. We can then use a combination of digital image analysis and machine learning to develop classifiers to predict outcomes when those drugs are used to treat those same tumors systemically. And we've shown that we can predict systemic treatment outcomes with 95% accuracy. We're now hoping to start a clinical study to validate this with actual patients.
This is Sydney again, here to introduce the Nmr Sensor project. Humans require water for life. Too much or too little water can initiate or aggravate a range of serious medical conditions. There is no current clinical gold standard for quantitative fluid assessment. Although many options exist, most are only useful for late stages of disease or are costly in terms of time, money, or equipment. The goal of the Nmr Sensor project is to design a magnetic resonance-based sensor to rapidly, non-invasively, and quantitatively assess a person's volemic status. We use the principles of magnetic resonance imaging to obtain a quantitative measure of water in a specified volume. A small array of permanent magnets creates a primary magnetic field, and a radio frequency coil transmits pulse sequences designed to capture signal from muscle tissue. By placing our sensor against the leg of a patient, fluid measurements can be obtained. In addition to redesigning the sensor and acquisition sequences to more sensitively measure fluid shifts, we will be testing the device clinically on dialysis patients. I'm happy to answer any questions about the Nmr Sensor project. Reach out if you'd like to hear more. Thanks.
Welcome to the Cima lab. My name is Kriti, and the project that I work on focuses on developing origami-inspired implants for local chemotherapy delivery into the abdomen for ovarian cancer treatments. This project is really special because it really spans the entire spectrum of translational research. We do everything from basic biology, trying to understand how this type of chemotherapy dosing that we achieve affects the body's immune system and native anti-cancer response, to device prototyping and design, all the way to treatment studies in animals. So it's a great way to get to experience translational research at all of its steps. We hope to see you next year here in the Cima Lab.
Hello, my name is Josh Murdoch, and I'm a member of the Nmr Subgroup of the Cima Lab, where I work on the design of an implantable biosensor device based on magnetic nanoparticles for continuous monitoring. Our clinical focus is on coronary care in the critical diagnostic window after a myocardial infarction. Our approach is based on the use of continuous dosimetry, similar to a radiation meter worn by X-ray operators, allowing us to pick up local concentrations of disease markers and store that information as an integral of total exposure through the elevations and remissions of temporally varying disease progression and treatment. Applications range beyond just medical diagnostics. Biomanufacturing, drug pharmacokinetics, and many other fields can benefit from having a continuous sensor with a memory. The technology we work with is magnetic relaxation switches, a nanoparticle clustering assay with a broad range of applications based on the self-assembly of complex particles around multivalent structures. This results in a change to the magnetic response of the sample that is indicative of the presence of the chemical of interest, readable by single-sided relaxometry and MRI. Feel free to reach out with any questions. Thanks for listening.
I hope you enjoyed this brief introduction to some of the people and projects of the Cima Lab. If you are interested in learning more about the group, don't hesitate to contact us.